So hopefully you remember when we had our overview of protein metabolism video, we sort of talked about when we're breaking down amino acids intracellularly, it doesn't matter whether they were exogenous or endogenous in origin, but if we need to be breaking these down, what we do is we're basically going to be snipping off the nitrogen piece to be disposed of in the urea cycle. So that's our elemental problem. And then we've got our alpha keto acid skeleton, and we're asking the question, who do you look like? So taking a little bit of a deeper dive on this, we're going to talk about how this problem is actually fixed. So in a very generic sense, we're going to be talking about taking any given amino acid, and we're going to be making it into its cognate alpha keto acid skeleton. Well, specifically, we're going to be talking about 18 different amino acids, and we'll talk about which 18 those are in a minute by talking about the fact that this chemistry happens by taking a different cognate alpha keto acid skeleton and converting it into its amino acid. So this is kind of an amine group exchange, if you will. That's not really what the chemistry is called. The chemistry is actually called transamination chemistry. So this is transamination. And the reason why I have 18 amino acids, because there's 2 amino acids that don't undergo this. And this is aspartic acid and glutamic acid. And the reason is, a way to think about this process is this really is sort of a funneling, if you will. It's a funneling of the nitrogen atom as that amine group of amino acids, not from 20 different amino acids, but we're going to funnel it into 2. So it's really kind of a nitrogen swapping reaction. And these are some kind of pairs I want you to be able to associate. Is the alpha keto acid skeleton of aspartic acid is oxaloacetate. And then the alpha keto acid skeleton of glutamic acid is alpha ketoglutarate. So if we think about this, what we're basically going to have is oxaloacetate is an alpha keto acid skeleton. It can interact with 18 of the other amino acids, or any one of those 18, and they're essentially going to be doing an amine exchange reaction. And then the oxaloacetate gets to pick up the nitrogen atom and become its cognate amino acid. It becomes aspartic acid. Whatever that other amino acid was, and giving up its nitrogen, it becomes its cognate alpha keto acid skeleton. So we basically take a list of 20 different amino acids that all have this elemental problem. They have that nitrogen there. And then we funnel it down, so we only really have to deal with 2. We have to deal with glutamic acid and we have to deal with aspartic acid. Okay? So here's the chemistry that we're really talking about here. Okay? Just sort of drawn in another way, we're going to have any given amino acid. So it's the same thing that I just talked about. We're going to kind of mention it and draw it in a different way. So we're going to be talking about the fact that this is any of those 18 amino acids, okay? And they can either become glutamate, or they can become aspartate. And just as a little side note, sometimes you'll hear me say glutamic acid, sometimes you'll hear me say glutamate. Those are the same cognate amino acid. The difference is really the protonation state. If we say glutamic acid, we're referring to it as a protonated state. If we say glutamate, we're referring to it as the deprotonated carboxylic acid, or the glutamate form. So unfortunately, I probably use them interchangeably, but important to highlight, we're really just talking about the same amino acid. That formalism really just has to do with what the protonation state is. Okay. So highlighting with both of these chemistries, we're basically going to have its cognate amino acid, so it's oxaloacetate to make aspartic acid, is going to be taking whatever that other amino acid is and making it into an alpha keto acid. And then on the flip side here, we're going to have alpha ketoglutarate being used to make its cognate amino acid, which is glutamate, and then whatever amino acid participated in that transaminase chemistry is going to make its alpha keto acid. Okay, so this chemistry is happening with transaminase enzymes. And I want to highlight an important cofactor is going to be vitamin B6, which is our PLP. We're not going to look at the enzyme active sites, but that is a required coenzyme that we need. We need to have vitamin B6 in order for this chemistry to happen. Okay, just as a way to kind of review some structures and think about things, I'm going to draw sort of the structures of alpha-ketoglutarate and glutamic acid, and then I'm going to draw oxaloacetate and aspartic acid, and we can see how this transamination chemistry works. So I'm going to draw amino acids a little bit differently than I normally would, but let's see, we're going to have... And I'm going to draw them sort of like they're related to the Fisher projections that we're going to see for our carbohydrate pieces. So we're going to have, here is our amino acid, okay, our alpha carbon with our hydrogen, and then this is the R group that we're going to have down here. So we've got 2 methylene groups, and then we've got a carboxylate group. So this is going to be glutamate, okay? So highlighting what we have for the R group is right here. So that is going to be our R group, okay? And what we're really looking at is what's happening with this carbon. This carbon is attached to a nitrogen when we have glutamate, which is an amino acid, okay? So this transamination chemistry let's draw what the structure is going to be of alpha-ketoglutarate, okay? So what I'm going to see is from the bottom, it's going to look the same. So essentially, we have this same R group here, okay? But now this carbon that was connected to a nitrogen atom now is going to be a carbonyl, okay? And then we're going to have, let me actually draw it like this. This might make a little bit more sense. This nitrogen is going to be converted into a carbonyl. Let me move this over, give myself a little bit more room here. Oops, didn't mean to erase that there. So that carbon atom is now going to be connected to an oxygen as a carbonyl. So we sort of have this piece gets swapped out, okay? So the carbon is going to be not connected to a nitrogen atom. It's going to be connected to a carbon atom as a carbonyl, okay? So this is, again, alpha-ketoglutarate, okay? So what I'm going to do is that I'm going to cheat just a little bit to kind of draw this. Copy and paste here. I'm going to do the same thing over here, but remember, what's the only difference between alpha-ketoglutarate and oxaloacetate and then aspartic acid and glutamic acid is a methylene group. So I'm going to delete that methylene group. Now I have made not alpha-ketoglutarate, okay? This is oxaloacetate. And not glutamate, I've made aspartic acid, okay? Otherwise the same. So hopefully what you see here is we need to sort of ask the question, looking at sort of the side chain that we have for the R group, we need to say, what metabolite do we have that looks like you? And one of the things that we're going to see at the very end of this video here is you tend to look like a citric acid cycle intermediate. So we're going to kind of connect in those points when we look at sort of a high level view of this, okay? So hopefully everybody's good recognizing what transaminase chemistry is going to be doing. What we're going to do and look at on the next slide here is we're going to ask the question, sort of, how does this then result in the disposal of this nitrogen? So to do that, I'm going to do another little copy and paste here. I'm going to copy and paste this piece, and I'm going to continue with this sort of on our next slide, okay? All right, so what we've really done is we've taken all 20 of our amino acids and the 18 that aren't aspartic acid or glutamic acid, we've done transaminase chemistry to basically funnel all of the nitrogen to either be aspartic acid or glutamic acid, and then we're going to have a collection of alpha keto acid skeletons from those 18 amino acids. Okay, so here's what we're going to need to do is we want to build urea. So here's the structure of urea. I'm drawing it like it's an amino acid, but it is H2N. Okay, this is the structure of urea. So what we're going to do is talk about how we build urea. We are not going to go through the details of the urea cycle, but building urea is what the urea cycle is about. So again, this is the urea cycle. Is all about building urea. So we're going to talk about how we build the different pieces. And I want you to think about building these different pieces of urea from sort of 3 different parts, okay? We're going to have this part. We're going to talk about where we get that part. We're going to have this part over here. We'll talk about where we get that part. And then we've got this middle part. So we're going to talk about where we get all of these parts. As a preview, it's going to be related to aspartic acid, glutamic acid, and bicarbonate. Okay, so let's talk about the chemistry that happens to do this. Okay, glutamate is basically going to undergo oxidative deamination. So oxidative deamination. And I don't need you to remember this specifically, but it is oxidative deamination. So we're basically going to have NAD P plus here. Something needs to get reduced, so it's going to be either NAD or NADP plus, generating NADPH. But then we're going to generate an intermediate, and you don't need to remember the name of this. This is alpha aminoglutarate. And we are going to do hydrolysis of this. Okay, so we're going to use water to hydrolyze this, and we're going to get alpha ketoglutarate back. And then we're going to get ammonium. We're going to get ammonium ions. So basically, we've taken glutamic acid and we found a way to selectively snip off just that single nitrogen atom as an ammonium ion. And that ammonium ion is what's going to be used to make one of the nitrogens that we're going to see in urea. So this guy is going to make this guy over here. So just kind of highlighting our colors here. This is going to make this guy here. It makes it from oxidative deamination of glutamate. Okay? So how do we get the other piece? There's actually a little bit more biochemistry that's involved in taking aspartic acid, but aspartic acid is ultimately going to donate its amine group over here. So again, we're not going through any of those details, okay, but part of the urea cycle to take and get that other nitrogen atom from aspartic acid, okay? And then the last piece to highlight here is a very common single carbon atom source for building things is going to be bicarbonate. So bicarbonate is going to be used to make that central carbonyl carbon. Okay, so all of this happens in the urea cycle. Ultimately, oxidative deamination of glutamate to generate ammonium ions that make that one nitrogen atom. Aspartic acid through the urea cycle is going to generate the other nitrogen atom, and the carbon and oxygen atom come from bicarbonate. Okay, so lots of really neat biochemistry within the urea cycle, but just highlighting here, I want you to know why we funnel things to aspartic acid and glutamic acid, and then the structure of the urea and how we take and put these pieces together. All right, so for this last little piece, I'm going to copy and paste this guy right here, okay, because I want to build on this, okay? So I'm going to copy and paste this guy over here because we want to ask the question, sort of, who do you look like, okay? And so ultimately at the end of the day, once we do this funneling, so I'm going to copy this guy here. Once we do this funneling, let me make this a little bit smaller so I've got some room. Once we do this funneling and we ask, well, what about these alpha keto acid skeletons? We're going to have 18 different alpha keto acid skeletons, and who they look like is going to determine how we metabolize them. Okay? One other little pairing that I want you to know. I want you to know oxaloacetate aspartic acid, alpha ketoglutarate, and glutamic acid. I want you to know alanine and pyruvate. So let's actually draw these a little bit differently so you can sort of see them in a different way. So we're going to have alanine. Remember, alanine here is going to have a methyl group, okay? So if we think about, again, swapping out this, let me draw it here, this carbon-nitrogen sort of group. Here was our R group. Okay, when we do transamination chemistry, let's draw pyruvate. So pyruvate is going to have that R group, and now we're going to have this guy as a carbonyl. Right? Here's our 3-carbon piece, so this took alanine and made it into pyruvate. Okay, still having our R group, and rather than having a carbon nitrogen, we've got a carbonyl that we have there. So another pairing I want you to know. Okay, so when we talk about these alpha keto acid skeletons, we want to think about what we're doing with them. And sort of what we have here are all, and we are not going through this in detail, but what we have are all different amino acids and identifying where they can sort of feed into the citric acid cycle as an intermediate. Reminding ourselves that ketogenic amino acids can only be made into acetyl-CoA. Glucogenic amino acids can be made into glucose. And if you are a ketogenic amino acid and can only be made into acetyl-CoA, there is not the option to be made into glucose. Okay, so the rest of this learning unit is going to be about stories of some of these amino acids. So I want to kind of highlight the ones that we're going to revisit in sort of future videos here. We are going to revisit the phenylalanine story, and we're going to talk about the metabolic disorder PKU. We are going to be talking about methionine metabolism and how it connects to MTHFR. So we're talking about folate metabolism, we're talking about methylation substrates, and so we're going to tie that in with methionine. We are going to be talking about glutamic acid and glutamate, and we're going to tie that into the story of MSG, right? And the flavor umami, and why we are hardwired to like the taste of anything that contains glutamate. Okay, case study that we're not going to necessarily cover, but if we do, we're going to be talking about things like isoleucine and leucine, which are branch chain amino acids. So branch chain amino acids, and we're going to talk about the importance of those if you are doing anabolic processes like building muscle. Okay, so the rest of the videos are going to be talking about some of these important stories like PKU and why anything that contains NutraSweet has a warning label on it. We'll talk about MTHFR. Remember, we learned about that last semester in our nucleic acid chapter. That's one of the important SNPs that impacts folate metabolism. And then we'll talk about MSG and its implications as a flavor additive.